The last thing that we need to be able to do is actually look at some actual data, right? And look at some changes in methylation patterns over time, or I'm going to have a question after this video about how methylation changes in cells that produce tumors versus cells that are healthy. And in this situation, what we have is we have a pair of monozygotic twins. Monozygotic means they came from one egg, so they're identical. They're identical twins. So in these two pictures here, we see a 3-year-old twin. I'm sorry, we see the twins when they were 3, and then we see the twins when they were 50. So what I want you to imagine is this is a chromosome from one of the twins, and this is a chromosome from the other twin, okay? And then this is the same situation over here, but 47 years later. So if we're looking at this, the first thing, if we're just comparing these, if we're just looking at the patterns, the first obvious thing is that they're definitely different colors. Like they're definitely less yellow as time goes on. So let's talk about what these colors mean in this situation. There's different ways to represent the change in methylation, but they're using colors here. So this the green color means hypermethylation, which means there's many differences in the methylation patterns between the twins. So green means there's more methylation. So in these areas, there's lots of differences. Red means low methylation. So they've actually gone lower between the two. And yellow means that there's no difference between the twins. So yeah, right, there's definitely been hypo or hyper, right? Hypermethylation or hypomethylation occurring over those 47 years. Also, if we look at the individual twins, we notice the patterns are slightly different, right? There's definitely different sequences. So they're, because of the environmental influences, and we don't know, I'm sure these patterns would be a lot different if they were exposed to different environmental factors, different stresses, different diets, all those kind of things. But there's definitely different changes here. So that's the skill they're asking you to do. They're asking you to first look at the evidence, make some comparisons, and then last, can you suggest some reasons? And if there were many differences between these two, and there are pretty good differences. Like I see a little yellow there, but not there. Here we have red, but this looks like it's all dark green, and then vice versa here. Here there's a little bit of similar methylation patterns here. Next thing they would ask you to do is look for the differences between the two individuals and then suggest reasons for it. And some of which I just talked about, stress, the activity we're going to do at the end of the week. Definitely has you look at how stress can affect how different genes are expressed through methylation or acetylation. So before we end this, I just want to talk about the question I have after this video. So just so you can kind of visualize this, there's a gene that is a tumor suppressor. There's also genes that activate mitosis. So there's constantly a battle in your cell whether to activate cell division, because if it's overactivated, right, that ends up cells dividing out of control, and then it can result in a tumor. And the other one is you've got something called a tumor suppressor gene, which is constantly trying to stop it. So it's these battles between these two genes that are trying to control your division. And whether those are methylated or acetylated can determine whether a person develops a tumor and perhaps even cancer. So that's what that question is about. So good luck.